Well, first of all, let me thank the kind invitation for me to come here to share with you some recent research progress, particularly in the areas of developing nanomaterials for two applications. One is for the selective extraction and sequestration of fine chemicals and fuel-related chemical from algae. And the second part is how we actually can convert various different kind of biological feedstock to biodiesel. And we basically using these mesoporous material as catalysts. So let me start by showing some of the pictures that you probably have seen in different versions, or maybe even this exact same one, that to emphasize that the importance of live in a sustainable society. So obviously that many of you have seen this kind of pictures that this is a glacier in Argentina. So you can actually see that in 1928 it's all frozen solid, and 2004 it's pretty much all the ice is gone, and you got the liquid form of the water. So obviously pictures like these are really the great reminder that one need to develop new technologies and also to be responsible for the society and for the future generations. So many people, of course, that will say that, well, maybe this is just some kind of abnormal phenomena happens somewhere else in the world. But until I saw the next slide, I think the debate is over. So I love this slide, by the way. So as you can see that, 18th century go all the way to 2006. And, well, I think that this hopefully will convince you, really, there is a global warming. We need to pay attention to this. And so with that, what does this mean to a sort of a simple-minded chemist and also a technology person such as yourself? That means that one should not just focus on using the petroleums or any sort of a fossil fuel as the feedstock to build a society. One needs to sort of perhaps learn the lesson from our ancestor that they utilize a lot of different resources around us. And then most of these resources are actually sustainable and also recyclable and being labeled as green. So, however, since that we live in a society that pretty much built all the things around us pretty much came from petroleum, your tabletop, your laptops, everything. So that is to say that we essentially, in the last 200 years, we have built a technology platform, a sort of infrastructure that is so designed to accommodate the petroleum-based type of feedstock. Now we're going to face a problem, a dilemma, that is a lot of these so-called green and sustainable resources are actually technically pretty challenging to the infrastructure that we have built. So do we want to spend another 200 years to build something completely new so that we can move from the petroleum base to the sort of a biorenewable base? And exactly what do we need to do in order to fully harvest these and utilize these resources? So in my mind, what we need is that the conventional chemical engineering type of the refinery settings that is based on the flow dynamics and things may not be quite applicable to some of these biologically oriented feedstock. That is to say, we need to develop a new refinery method for converting the non-food resources into feedstock. So that's one thing we need to do. The other thing is that, we pretty much, our whole society relies on using these sort of new materials that could serve as a catalyst to convert the feedstock to the useful the output. That also need to be redesigned, meaning the petroleum is actually a hydrocarbon. It's basically hydrophobic. You could build your pipeline with just a cheap metal, since that you're only going to flow the hydrophobic things in there. It's not going to attract the water. It's not going to trigger the corrosion. So you could actually build a pipeline miles long without worrying about these problems. Now you're going to switch to ethanol. You're going to switch to something that are actually quite attractive to water, to moisture. Then these pipelines, the existing infrastructure that you transport your crude oil from point A to point B, may need to be made by something else. So same thing with catalysts. Can the material functions at these challenging new interfaces, meaning water used to be you just need to work with the organic solvent, and in some cases, even gas and solid interfaces. Now you need to work with the water and solid interface. And so these are all new challenges. What can we do? So today, I just want to dive into one example and sort of give you a story that from the basic research go all the way to a private company to a pilot plant. So this is actually a very simple chemical reactions that I'm sure some of the chemists in the audience will appreciate. This couldn't be easier. I mean, I'm sure E will agree with me. This is what we call a transesterification reaction. That is, you take a triglyceride molecule, the structure drawn here. This can undergo a chemical reaction that convert it to another type of ester. And typically, if you use methanol as the reactant, this product is called the fatty acid methyl ester, FAME. And typically, the commercially known as biodiesel. And the side product will be glycerol, and the commercial name is glycerin. So this reaction, you just transform this type of ester bond to another type of ester bond. So just about anything that's strongly acidic and strongly basic can catalyze this reaction. So therefore, one could actually go, you Google biodiesel synthesis, you'll find a lot of recipe that they say you go to your backyard, get some methanol, get some sodium methoxide, you can make your own biodiesel. So why are we talking about this then? I mean, if it's such a low tech, problem. Well, obviously that, before I tell you the technical issue, I would like to emphasize that the reason why people think that biodiesel is actually considered to be a green fuel is for the following reasons. The most important thing I would like you to sort of take home with is that this is a biodegradable and essentially non-toxic fuel. And the other thing is that it actually, the most important part is that it could be directly applicable to the existing diesel engine, meaning that you don't need to build a brand new engines to accommodate the biodiesel. And then, the great lubrications and other properties are sort of the fringe benefits of this fuel. So this is all good. Now, if you look at the biodiesel industry, this is a map that I got from the National Biodiesel Society and National Biodiesel Board. You could actually see that in 2006, there were about 86 plants in the United States. And including Hawaii, there are actually 2. And so, obviously, suffice to say, most of the activities are in the Corn Belt in these I states. I'm from one, you're in the other. So this is actually quite interesting that why most of these biodiesel activities are actually sort of localized in the Midwest. But if you look at what happened now, this is actually most recent, June 22nd, 2009. You change the number to 173. So this is really a booming industry. And now you see more biodiesel plants being built nationwide. And this is where I came from. Even in the state of Iowa, the number had changed from 3 in 2002, now up to 42. So, and I'm sure similar trends is probably happening in Illinois and other part of the Midwest. So this is all nice and good. Obviously, that a lot of people think that, well, building biodiesel plant, maybe we can make profits. We also can create green fields that we can actually helping the society. So what are the issues? Well, as I mentioned, if you just look at the chemistry by itself, it's actually a very simple chemical reaction. But if you look at in reality, these are the necessary steps for you to convert the very simple triglyceride molecule all the way to biodiesel. So typically, you need to remove the water, meaning that you have to dry your feedstock. Your oil has to be dry. And if you're using some of the lower grade of the oil, you need to do the esterification. That's the first pretreatment reaction, followed by the transesterification reaction that I just mentioned to you. And oftentimes, you need to do a neutralization and a water washing step in order to purify the biodiesel before you can sell it to the market. And the side product are typically contain a lot of salt, and these glycerins are very low in quality, so you have to do some desalting to generate a high-quality glycerin. So all these issues is because one utilized these non-renewable catalysts. So the commercial available biodiesel catalyst is sodium methoxide or sodium hydroxide. So everybody knows that this is an inexpensive chemical. So the problem of this chemical is not only it's highly toxic and corrosive, it's a strong base. The real problem is this thing dissolves in methanol and stays in the methanol. That is to say, once you finish your reaction, there's no way for you to get this corrosive things out of there unless you do some neutralization by introducing acid. Then, of course, you generate a lot of salt. And that's why the water washing is necessary to remove those things. And all those things get moved down to the glycerin and water phase. And that makes the glycerin very low quality. And so you can't really sell it directly to the personal healthcare industry. And the other thing is that you have a catch-22 situations that biodiesel is an ester. The more of the water that you use, the easier they get hydrolyzed. They get decomposed. So you have to dry them. And another thing that sort of the DOE label as a sort of one major concern about the biodiesel industry is that at this point, 1 gallon of biodiesel production requires up to 8 gallons of water for washing. So this water consumption ratio is not very green. And so all these are actually the technical issues that need to be overcome. And, well, obviously, as I mentioned, it seems like most of the problems are sort of centralized to the usage of sodium methoxide as the catalyst. Okay? Another problem that I'd like to point out, it's actually a study that recently published by NREL. This is actually June 2008. One can actually see that, again, we're labeling all these biodiesel production facilities in the countries, this time by the feedstock, by the oil that they use. So the yellow triangle and the open circles are those that are using vegetable oil as their feedstock. So suffice to say, that's the majority. So the reality right now in our country is that the U.S. produces biodiesel from vegetable oils. So setting aside this fundamental argument about food versus fuel, we have another problem, that is the profit margins and also the amount of biodiesel you could produce is pretty much locked in to how much of the biological and vegetable oil that you could produce from these different areas. So why not use something that's non-food based? Use something that's actually, perhaps waste. So obviously a lot of people are investigating and see whether one could actually use animal fat or algae or even your restaurant oil as the feedstock to make biodiesel. There is a problem. Most of these lower grade of feedstock contains a lot of free fatty acids. That is to say that a few percent of the free fatty acids, it's enough of the acid to destroy your base. The acid runs into base and form a lot of soap. So the sodium methoxide is incompatible with those oil feedstock that contains more than just 1 or 2% of the free FFA. So that's why one needs to do a pre-treatment reaction to use an acid as the catalyst to esterified the free fatty acid into the biodiesel first. It's kind of converting this harmful impurity into the product first. Then you get this kind of a purified oil. Then you run the transesterification reaction with the sodium methoxide to give you the biodiesel. This adds a layers of the capital investments and another layers of chemical cost for you to get your end product. So what if we could actually find a material that does not dissolve itself into the solution, so could circumvent this homogeneous problem that I just mentioned of sodium methoxide, but yet the solid material could do both these esterification reaction and the transesterification. That would be ideal, right, to solve this problem. And another thing I would like to point out is the fact that another disadvantage of using vegetable oil as the feedstock is that if you look at the oil amount that you could produce from per surface area, if I could just basically drive your attention to the top threes that are these most common thing that we know in the US and the Europe, corn, soybean, and canola. If you look at these numbers, right, this is basically a table that I've taken from this review article, and so the unit is actually in the sort of eccentric system. So it's liter per hectare. So suffice to say, if you look at these numbers are not very high. So right now, U.S. use about 970 million acres for crops and grazing. That is to say, if you're going to combine all these numbers and do a back-envelope calculation and see how much surface area we will need if we're just going to use these three feedstock to make enough oil to give us the biodiesel to meet the current transportation needs, we are coming to a shocking conclusion. That is, about 16.4 trillion acres of soybean will be needed just to supply the current U.S. transportation diesel fuel. So everybody knows that if you just compare these two numbers, we know this is a pipe dream. This is never going to come to the reality. But if I could drive your attention to the microalgae, the bottom of this table, one recognizes the amount of oil that could produce per surface area from algae versus from soybean. Essentially, these microalgae are so oily that the amount of oil you could produce is in orders of magnitude higher than those of these traditional agricultural food-based feedstocks. So if we just do the same kind of calculation, what would be the amount of the land that's needed? If we could fully harvest all the oil that's produced by the microalgae, only about 20 million acres of the current land is needed. And that's, if I do the translation, that means only 2.2% of the existing crop and grazing farmland in the US will be needed to give you enough of the fuel to meet the current transportation needs. So this is great. However, well, before I go into the problem, I'd like to say one more thing. That is, well, essentially you know that algae, some of the algae are known as palm scums, right? So you don't really need a prime land. You know, you don't need the sort of a prime agricultural land. Basically, we have a lot of, retention ponds or lakes or river area can be utilized to grow algae. So this is all nice and good. Well, so can we actually find some wild type algae that are containing high enough of oil? Turns out we can very easily. Like the Batrachococcus brownie is actually a string of algae that's known to grow in the very cold climates. So in fact, in England, there's a lot of BB that's actually being currently cultured for the oil production. And some of these actually freshwater and the marine species are also pretty oily. So that's finding the wild type algae to grow enough fat is not an issue. So what are the issues? If you look at the chemical structures of the algae, one realizes that these are actually single-cell organisms, and some algae, they tend to form colonies. So for example, I'm using the EM image of the BB to highlight the point that here's the cell body, and they have the cell wall structures. And essentially, these single-cell organisms are quite smart. They store their fat, in their backyard. So it's between the cell membrane and the cell wall. But because of this intact cell wall structures, it makes the extractions of these oil pretty challenging. That is to say, you have to break down the intact cell wall in order for you to gain access to the location of the oil. But regardless, I mean, there is a tremendous amount of excitement, both in the government and the research area, but also in the private sectors. So if one just look at the recent investments that have been done by either the companies or the venture capital worlds, one noticed that these are actually individual algae-producing companies. So for example, if I could drive your attention to the synthetic genomics, we recently received an investment of $600 million from ExxonMobil. And by the way, the money is not coming from coming out of the R&D budgets of ExxonMobil. It's corporate money. So essentially, to ExxonMobil, this is just a one-time gift to this algae company to make biofuel. So most of these companies are actually focusing on identifying and adjusting the growth conditions so that you could produce high amount of lipid and also minimize the amount of the extraction challenge to give you enough lipid to make biofuel. So the projections to the 2025 is that we will be able to produce about 6 million of the gallons of algae-based biodiesel to meet the world needs. And so this is all very exciting. And the current approach of growing algae for the biofuel productions can be divided into two major categories. One is the so-called open pond approach. So essentially, you will have these pedal wheels on the open ponds, and basically this offer the aerations and also offer the circulation of the algae so that these algae can expose to the sunlight. And so you see a large activities that, for example, this is the Cyanotech corporations in Hawaii. You can actually see that essentially the whole peninsula is being converted into an algae farm in the open pond approach. And there's another approach, for example, General Atomics and Synthetic Genomics and Solix. They believe in using photobioreactor. So Essentially, you have plastic bags or test tubes or the reactor that you can actually encapsulate the algae and grow the algae inside these tubes, and providing the artificial solar energies and the CO2 to keep them under a precise control. And Solex's vision is to build a farm of this kind of bioreactor in the desert. They just started their first projects with a, I forgot what's the location, but it's the southern part of Colorado, and they basically have signed a contract. Right now they have a 2-acre farm of using this strategy. So if I want to sort of tell you how we actually could grow the algae and convert it all the way to biodiesel, I feel that this is probably the best cartoon to highlight the point. So as I mentioned, most of these algae growing companies, they're over here. So they're selecting the right strains, they're growing the algae, and then they have all different kinds of the IP-related technology allow them to starve these algae from nutrients so that they could produce maximum amount of fat. And then they typically will just use the brute force approach to actually crush these algae and isolate the lipid. And some are actually using the oil wet milling technique. For example, they could use the hexane to extract the hydrophobic oil compound from the algae biomass and then take it down and then do the further processing. But I only would like to point out one thing. That is, when you mechanically or chemically destroy these single-cell organisms, you don't just get the fat out. You get a lot of compounds that come out sort of almost in the situation that you have an alphabet soup. And only certain chemicals in the soups are suitable for the conversion to produce fuels. To give you the precise idea that algae lipid, we're not talking about the charged lipid. We're only talking about the neutral hydrocarbon lipid. It's a very complicated mixture, starting from your ether lipids, the triglycerides, to fatty acids, to terpene, to alkenes, to sterols. Okay? So obviously, all the algae producers will tell you that this is not a problem. This is fantastic. Why? Sterol is actually a great nutrient. Fatty acids, some of the fatty acids, you know, the omega-3s, are actually great. So these are actually fine chemical, value-added compound that could produce from the same algae culture. But they never tell you is that how can you economically go in and isolate these different categories of compound and then to make profits. That is actually the most expensive process and the challenge that's facing in this industry. The reason why that's a problem is that, as we know, only those short-chain, non-branch hydrocarbons are economical feedstock to make diesel-based of hydrocarbon fuels, gasolines, and so on and so forth. So if you have the sterols, which is a heterocyclic compound, you don't really want to burn that because it will be incomplete and generate a lot of problem. And phospholipid is yet another big problem. And the free fatty acid, as I mentioned already, is going to destroy your sodium methoxide, so you can't make the biodiesel out of that. So how could you extract these oil out from the algae is currently a challenge as well. If you crush them, obviously that means you kill them. So or if you use the hexane as extraction, these are also lethal and fatal. You'll cause the lethal and fatal damage to these to the cell growth, meaning that you have to kill the animal in order to get the valuable compound out of them. Another big problem is that there is no efficient and economical refinery method to isolate the suitable fuel feedstocks from the alphabet soup of the hydrocarbon, the algae. And that I already highlighted in the previous slides. And so the most important challenge is that if you don't isolate those right compounds, if you directly charge your so-called algae oil to your biodiesel reactor using sodium methoxide, the free fatty acids, and all these other impurities will destroy your catalyst, and you cannot successfully get the end products. So all these are the current technology challenge. I believe that the nanotechnology could play a role in helping to sort of circumvent these current challenges. So today, I mainly would like to focus on how we can actually develop the nanoparticle base of a material serving as absorbent to help you to isolate the right alphabet from this alphabet soup. And also would like to show you how one could develop a solid catalyst that allow you to efficiently convert these oil to the biodiesel. So let me start by telling you our basic approach. We'd like to develop a non-invasive extraction methods with high recyclability and regrowthabilities of algae. That will be our long-term goal. If we could accomplish that, then we don't have to kill these animals. We could actually even have a smaller footprint, meaning a smaller pond, if we could recycle and regrow these algae repeatedly. And we also would like to develop a new sequestration method for the isolations of suitable fuel feedstock from the hydrocarbons of algae. And the last part I'll focus on is we have developed a solid catalyst for very efficient productions of biofuel from these different oil feedstock, with the fatty acid or without. So let me first tell you our basic approach. So obviously we're facing a sort of a challenge that the sodium methoxide, even though it has a lot of problems, it is actually dirt cheap. It's about 5 to 7 cents per pound. And so for 1 gallon of biodiesel production, at best, the cost is about 5 cents. Okay? So therefore, how can you come up with a nanotechnology that could be price competitive to solve this kind of a huge commodity problem? So our group specializes in making the sort of a nanoporous material by using surfactant micelle as a template. And I'm showing here a cartoon that basically this is a CTAP surfactant. They will self-assemble into a micelle in aqueous solution. And one could actually introduce silicates. Then these things, silicate, will cross-link and form sand, meaning silica, around this organic templates. What we do is we introduce a very different kind of chemicals. For example, this is the organosilane. This blue ball here will have the ability to compete with the silicate. So this molecule will know how to orient itself, and then the other end of this molecule has the same kind of substituent as the silica. So later will be incorporated covalently to be part of the structure. We could use this sort of a simple one reaction in situ type approach to make a range of different kinds of material. To give you a sort of a more detailed explanation, but a sort of a simplified scenario, is that if one throw in an organosilane, where this R group here is hydrophobic in nature, obviously you have a situation that in the water you have these micelles that have the hydrophobic core and the hydrophilic interface. The best way for this molecule to hide the hydrophobic moiety from exposing to water will be to intercalate into the hydrophobic core of your micelle. This, of course, will change the curvature of your surfactant assembly, and also sets up the scenario that this layer of silicate is very close, and so that the following reaction, meaning the condensation reaction to form the SiO-Si bond, will be very, very fast. If you go to the other end of the spectrum, where the R group here is hydrophilic in nature, then that incentive is gone, meaning that for these silicate to find each other, this molecule will have to tumble around to find the right angle to form the silica wall. So using the slow condensation and fast condensation, I'm happy to report we could actually make a whole range of different particles with the organofunctional group already embedded to the structure. But the most important thing is that we could tune the structures and the particle morphology, meaning the shape and the size of these particles, anywhere from the tubes to the tube, fan noodle, spheres, raspberry, bars, and so on and so forth, with a precise control of the surface area and the degree of functionalization. And one afternoon, a high school graduate student can make actually kilograms of this. So it's not really your sort of a high moisture sensitive dry box type of a chemical synthesis. So shown here is actually 2 TM image. To your right, we're looking down the pores. Hopefully you can see those light colored dots representing the openings of the channel. So essentially what you see here is like it's a miniaturized of honeycombs. And this is the side view. Hopefully you can see those stripes. They're packed in a parallel fashion. So literally what we have is a honeycomb that is being shrunk down to a tiny little sphere. And this particle here is about 200 nanometer in particle diameters. So this particle is even in the size windows of virus. And the reason why we like about this material is that the surface area of this material, 1 gram of material will give you over 1,000 square meters. So 1 gram a football field. So we actually have used this kind of a porous material as a platform to do a lot of catalyst design. So we now have a complete controls of morphologies. We could actually put these different kind of organofunctional group there and ask these organofunctional group to help each other to do some sort of a cooperative catalysis. So in essence, this is sort of like a tiny little enzyme active sites that you actually now have these functional group are helping each other and in catalyzing the reaction. we also can actually make mixed oxides, meaning that this material is not just made of sand, but made of different kind of metal oxides. And today I'll show you exactly this example to tell you why this material is actually effective and a good biodiesel catalyst. Another thing, before I dive into the catalysis part, I'd like to mention is that because we could truncate, we could actually miniaturize these particles, and we have the ability to functionalize the surface of these particles. Turns out these particles have a great membrane permeation property. That is to say, we can actually use this to build a Trojan horse, to hide drugs inside the pores, and we put things on the outside, for example, exogenous genes, and this particle can be internalized by animal cells and go inside the cells, and the drug could do drug delivery, gene could do gene transfection. So there's a lot of application in the biomedical and the biotech area. But today, I mainly would like to focus, to sort of recognize the difference between animal cells and these sort of algae and plant cells. The main difference is that these plant cells have the intact cell wall. So this is actually much more difficult to penetrate than, let's say, just to being endocytosed by the cell membrane. So in 2007, we actually have developed this catalyst, this particle, should I say, that by covering the outsides with carbohydrate, this particle is now small enough and lubricated enough to penetrate the cell wall and go inside the plant cells. So this is basically the image of the show. Well, I guess the resolution is not that great, but hopefully without the coating, all the particles are being excluded on the outside of the plant cells. With the coating, the particle can indeed penetrate the cell wall and go inside the plant cells. So represented by those green fluorescent dots. And the red fluorescent is actually the chloroplast of the plant cells. So using the strategy, now we could actually deliver things in there. We could deliver a plasmid genes in there and trigger the gene transfections. So a large plant species, such as the tobacco leaf, can be transfected and could actually express the high densities of green fluorescent protein. So that's for the highly developed plants. What about algae? What we found is that if we just take algae cultures, the image is shown here. This is, again, BB. When we actually culture them in the test tube, such as this, one could actually identify a lot of these green colonies. When we sprinkle the particle, such as this one that we create, this is actually a carbon base, not a silica base of particle. Immediately, all these particles gone after these algae colonies. So you can actually see all the algae colonies being covered by these green particles. And they actually absorb a lot of fat. Okay? So the indication is, this is a sort of complicated slide, so I'll just walk through this real slowly for you. Essentially, we isolate the particle, and we could actually wash it with hexanes and water. And those that could be removed from the pores, obviously, are the one that's not strongly absorbed by the particle. Then we analyze the particle and see what kind of fatty acids are being sequestered by the particle up here, shown in this spectrum. Compare with the hexane wash and the water wash spectrum. So I just want to highlight those that shows up exclusively only in the particles. Those are the carbon-14, carbon-18, and carbon-16s. Okay? There are some of these black labeled the fatty acid peaks that shows up both up here and down here as well, meaning that these are the fatty acids that's weakly absorbed by our particle. And then you have some additional ones, for example, these large waxy fatty acids that are not being absorbed by our particle. So a translation of this is that these short-chain, straight-chain fatty acids are actually strongly absorbed and being sequestered by our particle, whereas these ones are actually weakly absorbed. And the waxy, that are non-economical fuel feedstocks, are not being absorbed by the particle. So we could also develop different types of a silica-based particle and just pack them in a column. We could flow the hexane extracts or different kind of organic extracts of lipid from algae, directly flow through this column, and we can analyze what's in the filtrates and what's been absorbed by the particle. Turns out, our particle has a very high absorption efficiency for a lot of different natural fatty acids that are created by algae. So as you can see, that the lowest one is actually already 85%. And so this can be done in a sort of large column type of setting that allow you to actually marry this to a lot of different commercial available algae lipid extraction technology. Now I'm going to sort of shift gear to tell you a little bit about the catalyst design. So as I mentioned, the free fatty acid is actually one of the main problems why you need to do an acidification reaction that's typically catalyzed by an acid catalyst followed by the base catalyst to give you the biodiesel. And we have initially our thinking was that, well, since that we know how to do chemistry inside the pores, we could take advantage of the two different kinds of surfaces of this material, meaning exterior particle surface versus the interior pore surface of a particle, to tackle this problem. Initially, we thought that we could basically just put the acid function group on the outside and put the base function group in the inside. So their acid and base are separated from each other. They will not kill each other. So this naive thinking is why we start designing the system, that we have the free fatty acid without going through a diffusion into the pores, interior pore. They could directly be converted to biodiesel on the outside. Since they're only a few percent, then the rest of the triglyceride could take its time to go into the basic side, being converted to biodiesel and glycerol. So the big challenge is that, well, what kind of things that you can introduce as a catalytic moiety without paying a huge amount of a price, because sodium methoxide is really dirt cheap. So we decided to use some sort of natural minerals, for example, calcium oxide. We could actually introduce the calcium oxide to the assembly, the synthesis of our nanoporous particle. Initial thinking is that these calcium oxide particles could serve as the basic catalytic domain. They're kind of like little marble that embedded to your amorphous wall, so that these green spots can serve as the catalytic site for you to convert the triglyceride to the biodiesel. Then we would like to put some acidic side on the outside to do that. But much to our surprise, once we finish the synthesis of this basic calcium oxide functionalized silica material, we did the catalytic conversions of various different kinds of the feedstock. So I'll just first start with the soybean oil. Turns out this catalyst is very, very effective. You only need about 8% of the catalyst per oil weight ratio. And the methanol, you could use very, very small amount, only 0.16. This whole operating temperature is actually refluxing methanol and then open air. You can actually quantitatively convert this to biodiesel, and with a high recyclability, more than 20 times you can actually recycle these particles. Simple filtration, fish them out of the reaction, do it again. So New York Times was pretty, they came to us and then they did the story on this catalyst development technique. And, but the most sort of a surprising thing scientifically was from these two plots. So we were not surprised that our catalyst, given it's heterogeneous, given that it's quite stable in chemical structure and reactivity, we were not surprised that this catalyst can be recycled and reused more than 20 times for soybean oil without sacrificing and seeing any decrease in the reactivity. But we were very much in shock when we did this poultry fat testing. This is a poultry fat sample, the chicken fat sample that we got from Tyson, containing about 18% of the free fatty acid. So since we're only using 8% of the catalyst, we thought that for sure that 18% of the free fatty acid in this feedstock will destroy all the catalysts that we introduced. But we, in fact, could actually recycle and reuse this catalyst more than 8 times before it gets deactivated. So the question now becomes, we understand that the calcium oxide is a basic metal oxide that could do the transesterification reaction. But silica, even though it's a weak acid, is certainly not a catalyst. Otherwise, we're in trouble, right? I mean, sea sand will catalyze these reactions and destroy our ester groups in our body. That's going to be a chaotic situation. So we know silica is not acidic enough to catalyze the esterification of the free fatty acid. Then the question becomes, how come the free fatty acid can be converted to biodiesel? And this free fatty acid did not destroy the calcium oxide domains of our particle. That was our scientific question. So how did the esterification of free fatty acid happen? We started to analyze the structure. This is actually commercial available calcium oxide. This is the crystalline domain X-ray diffraction patterns. This is our synthesized material before we do the calcination, before it activates it. But suffice to say, if you compare the diffraction pattern and the location, meaning the 2 theta positions, this is not this. Meaning our initial hypothesis of that embedding these crystalline domain of calcium oxide into our silica matrix, this hypothesis is completely wrong. There is no crystalline calcium domain in our materials. So can this be some kind of mixed oxide? That was our first question. So when we come to talking about calcium base of the mixed oxide, one naturally will think about cement. Portland cement is actually a natural mineral that contains 60% of the tricalcium silicates. The other 15% are actually beta dicalcium silicates. Both these calcium silicate mixed oxide will undergo hydrolysis. That is to say, they will generate this hydrated layer. Okay? And typically people call them CSH layer and the calcium hydroxide. Same thing will happen to the dicalcium silicate. So if we zoom in and take a look at the structures of this hydrated calcium silicate layer, one finds that it actually has defined nanostructures. So this is typically called the tobamorite. It has the chemical formula of this, and this is actually the pictorial drawings of how the structure looks like from the top to the bottom. And if we turn this 90 degrees, it will give you a better view. So essentially, the light gray polyhedra are actually the calcium oxide layer. So on top and below, this calcium oxide layer is covered by only one layer of a silicate, represented by the tetrahedra. Then this basic building block packs on top of each other like lasagna, layer on layer. The interesting thing is that the silicate, represented by these tetrahedras, are not touching each other, meaning that these teeth are actually not connected. There's a gap between these layers. The gap is filled with water and other kind of counterions. So calcium oxide, silicate, gap, silicate, calcium oxide. This is the basic building block of this of cement. If we start taking a look at the XRDs of our synthesized material, one could see the striking similarity between the diffraction patterns of our material versus the hydrated cement. They're almost identical. So did we just spend the most, all the effort making the world's most expensive cement? So I told my student this is a concrete result. And it turns out, when we calcine, when we actually do the surface chemistry of this starting material, one could actually change the structures of this kind of hydrated cement structure to something look like this. MCS1 is the most active catalyst that we prepare. And I will not bore you with the sort of detailed analysis, only to tell you there are certain peaks that remain and certain peaks disappeared. So the take-home message is that, I'll skip this slide. This is basically the NMR analysis of that same structure. I'll just tell you the cartoon, what exactly happened. So as I mentioned, in cement, in the concrete world, this layer is actually filled with water and other kind of counterions. So the binding affinity comes from this kind of a layer. What we did is that we changed this gap. We start closing in on top of each other until the silicates, meaning these teeth, start to connect. So we transform this kind of a silicate structure to this kind. So if you actually take a look of these, when they are freely separated from each other, basically the silicon oxide here in this tetrahedral, the silica is connected to two of its own kind. Now when you connect, you transform the so-called Q2 to Q3. What we found is that more of the Q3 that you generate, the catalyst seems to be more active. Okay? So our catalyst is structurally similar to cement, but not cement. Okay? So using the sort of spin counting result to give you indication, if you have sand, if you just take sand, you start putting some calcium on the surface, you're in this regime, which means that the calcium and silicon ratio is very low. You got mostly silica, a little bit of calcium. This is just sand. Okay? If you're over here, okay, when you reach the 1.7 ratio, seems to be the most active catalyst that we get. If you go higher than this ratio, you're in the regions of cement, and that's not very good in terms of reactivity and recyclability. So turns out, because of this kind of a situation, you're forcing the calcium into a silica coordination, this mixed oxide coordination. You are actually creating a charge difference. So meaning that the silica that force into the calcium oxide binding domain exhibit a higher Lewis acidity, meaning that those silica now plays the role of an acid catalyst, where the calcium oxides are playing the role of a basic catalyst. So essentially what we have is a material that could behave both acid and base for the two reactions that of our interest, which is the esterification reaction of free fatty acid and the transesterification reaction for the oil. And that's how we rationalize this reactivity. And so because of this results, we were lucky enough that we were funded by a venture capital firms that's outside, right outside of the Stanford campus. It's a pretty big venture capital firm. They have about $20 billion a year. They have about 56 portfolio companies. We're one of the 56. So they gave us about $12 million for us to establish a company called Catalin. Initially, I would like the spelling of C-A-T-A-L-I-N. It's the combinations of catalysis and lin. But turns out that spelling was taken by the Swedish shampoo. So now I have to sort of settle for the second best at the C-A-T-I-L-I-N. But I still like to pronounce it as Catalin. And so essentially, using our solid catalyst technology, we could actually now streamline and eliminate some of the key steps in this sort of a traditional biodiesel process. So every circle and boxes representing the key step of the traditional biodiesel process, we could actually eliminate a lot of them, okay, and just introduce the three new components, meaning the solid catalyst, a filter, and a dry wash, okay. So the take-home message is that we could actually lower the capital cost by about 50%. Okay, meaning that all the acid, pretreatment, water washing can be gone. And we could save at least 10 cents per gallon. And our catalyst is made of inert element, as you know. And so it could actually be reused and recycled. When you're done, landfill would be the way to dispose them. So right now, we are commercializing this catalyst, and shown here. We just finished the synthesis of about 2 tons of this catalyst, and we're now doing beta testing for several biodiesel producers in the country, including REG, that's really right, our next door neighbor, and also several biodiesel producers, one in Houston, the other one in Louisiana. And so this is the current catalyst, the Catalin team and myself, and these are actually the 3 ladies in the front rows are actually my former students and postdocs. And Wayne Turner is our VP for operation. He was actually with Dow Chemical. He took an early retirement and joined the company. The CEO is Larry Lenhart from the Moore Davidoff Venture, and Dave Sams was actually with GW Grace and also with Shell Chemical. And so he joined us for the VP of business. So we have an R&D lab in our incubator space. We also have a pilot plant in this place called the Bioenergy Conversion Center. This is this is actually our 1 million gallon pilot plant. And basically, everything is fully automated. Only 2 operators needed to run this whole facility. So with that, I'd like to thank the name that I highlighted in blues are the one that contributed to this project. And the money for the research mainly come from the Department of Energies, from the Basic Energy Science for the nanomaterial synthesis, and ERE for the algae. And for the material synthesis, there's also money from the NSF, and USDA, and also Pioneer for the gene transfections and fine chemical sequestration from algae projects. And this is my current group, so just want to circle the 2 guys. Brian Truans contribute a lot to this, and also Tommy Chen, also is the key players of this research. So with that, I thank you for your attention. I'll be happy to answer any question. Thanks.
Yes, sure.
So when you talked about the nanoparticles penetrating the cell wall, 2 questions come to mind.
Sure.
One is, obviously, then you cannot be introducing this willy-nilly into the open contact system, so that would be one kind of question. The other is, are all, say, if you're using it for algae in particular, are the different species of algae, do they have very different characteristics? Does the particle penetrate the cell wall with different types, or only certain types are amenable to this kind of treatment?
Sure. Let me answer the second question first, and I'll go back to the first question. So we are now currently only interested in interfacing our particle with the wild-type algae. So right now, we have about 5 or 6 different kinds of algae growing in-house, but we're actually collaborating with algae producers out there. suffice to say, those high lipid producing algae are those that contains mostly intact cell walls. Okay, there are only one or two that produce enough of the lipid, meaning around 30% by weight, yet contains partial cell wall. Okay, so therefore cell wall penetration is a key challenge right now. Okay, but there are algae that contains almost no cell wall or only partial cell walls. So we are actually quite interested in interfacing our particle with those. And the idea is that if you have the particle goes in and get the fatty acid out of there, you hijack their metabolites. How would these living cells respond? Would they respond in the way that, okay, my precious chemical just got stolen. Do I need to make more in order to stay alive? If that can be realized, then you have a true nanofarming going, meaning that you could upregulate that metabolic pathway. Okay, so that's long term, right? Right now, our particle initial idea that we want to do is to do it inside the photobioreactor. So your assumption is correct. But we were also contemplating in the sort of a control open pond environment, can these particles, by absorbing enough of fat and fatty acid to make them hydrophobic enough, they float on top of the pond so that one can do the skimming. Okay? But right now, we are not there yet. Right now, we could get them in. They absorb a lot of fat, but they don't come out. They form agglomerations. Just like any hydrophobic thing in the water, as you know, right? Once they load themselves up with fats, they just form agglomeration. We essentially see a tumor inside of these algae. So,
where they do a similar kind, I think they propose this kind of a farming concept as well. But I believe, if I remember correctly now, they were using a two-phase system using dodecane as a milking agent.
you're talking about phytokelp type things.
I'm sorry?
There is a company called Phytokelp.
Oh, is that what it's, okay, I read something about
But actually the milking, you're talking about milking process, right, in Netherlands. And so the milking process is different. So the milking process is that you make a layer. You sort of essentially do it by phasing. I think Lance is probably very familiar with this. You have two-phase system. You have a organic solvent. You could actually use different kinds of things, and hexanes or fatty alcohol, so on and so forth. You have the water underneath. You have a bubbling system. You could actually somehow trigger the mixing, and then the lipid will be extracted into the hydrophobic. The key thing is the mixing, is the resonance time, right? If you have an intact cell wall, and if that cell wall is actually depends on the cell wall structures. The short contact time, of course, will minimize the invasiveness of this process, but suffers the low extraction efficiency, right? So then how do you find the balance of these things? It's another ongoing research. So there are actually different kinds of approach. The reason why we would like to use the particle system is that it's for separation purpose, okay? But regardless whether it's milking or this, one still face one issue. If you use this kind of milking process, right, you're not only getting your fat and fatty acids out. You're also getting your sterol, your chlorophyll, all these hydrophobic compounds can be sequestered by these organic solvents. Then how do you separate the good chemical from the bad chemical, quote unquote? Okay, so I think that this kind of extraction and sequestration technique is still needed.
In nature, alpha populations tend to bloom and then die off, another
Yes.
population comes up. Now, is this going to be a problem for large-scale biodiesel?
Oh, there is an extensive discussion about, so essentially that, well, of course, you have different seasons, right? I mean, so the algae will grow to reach the sort of maximum in the summer, and then, they will go to the dormant stage in the winter. And so how do you adjust that, and which species do you use? And right now in the industry, most of the people are the big algae growers. I shouldn't say all of them. I should say the big major algae growers. They seems to be more interested in the marine type of species than the freshwater. Okay, because it seems to be a little bit easier for them to stress those marine algae to give more lipid than the freshwater ones. The freshwater ones, they seem to be able to resist most of the stress factor. So that's my observation. But that is a big factor. One needs to consider that. There's a lot of issues. If you do open pond, evaporations of the moisture will change the salt content inside your culture. Some of them may die. Another issue, pathogens. E. coli like these greasy meal. I mean, we like them because of the oil, so would the bacteria. So that's another thing. And mosquitoes, not to mention.
One of your last slides, you showed some of your partners,
Yes.
including Crown. I understand their role, but you also showed Lurgy. What's their role?
They're actually now want to sign agreement with us and use our technology, being their next generation of biodiesel plant design. So they want to license this technology. And they did the reason I showed it on that slide is because they did the techno-economic analysis and to see, so using them to show that, okay, this lowering the 50% capital investment is being vouched by some of these engineering firms. Okay.
You show the 20 time cycle.
Yes.
Without any losing the ability, at least in the oil case.
Right.
What happened to 21? I mean,
It kept going, right? It kept going. The longest time we have tested is up to about 42 times. Then we gave up because every time you do this, you lose callus. So we started with 1 kilo and you're down with, because no perfect filtration system, so therefore you lose some of the callus. But depends on the feedstock. If the soybean oil, 42 times, no problem, but you do some of these animal fat or you do in a blend, then these things will still get deactivated, right? But we're in the business of selling callus. We really don't want our callus to live forever.
That's not very green.
I should slap myself. Oh, yes. Yes.
So poultry fat is usually solid. Is it solid to solid reaction, or do you do?
No, if you elevate it to about 65 degrees C, they melt. Right.
So it's not like the fat you buy from the
Typically, right now, the large biodiesel producer, they call it industrial oil. They typically will blend like tallow, poultry fat or something. So they typically will blend them into an oil feedstock because that to them is the most economical way. Because as you are correct that most of the animal fat at room temperature they are solid, and the chicken fat that we got, essentially what they did is that they just basically after trimming off the things, they do a sort of huge French press type of things, and then whatever flow on top of water, they collect that. They call it chicken fat. So there is protein, blood, feathers, things like that in there. It's very disgusting.
Anyone who has to leave to get to class or another commitment, this would be a good time to leave, and then we'll continue questions as long as Dr. Lin is comfortable taking questions. But it's good to let people go without creating a disruption.
Sure. Yep. Nice. Yeah. Yeah, thanks. Yes.
One of your slides showed they produce the oil in Hawaii along the coast. Isn't it toxic to the resident? I mean, a lot of algae have the cyanotoxin.
Absolutely.
That is not very good.
That's right. So I believe they are basically that. I think what they're doing is either Corolla or Nanocoropsis. Those are the two that they are actually doing the cyanotech there. So I think that they have a wastewater treatment plant right next to that facility, and they basically, they're doing that basically to process their wastewater before they directly charge it back in. But I think the water inlet is directly from ocean, so it's coming in but not discharging it directly. So I think they minimize the environmental footprint by having a wastewater treatment plant. Right.
The breeze will bring in the toxin to people.
Right. I don't know the level.
Yeah.
I don't know the level, but that's a good concern.
Yeah.
But that thing has been going on over there for at least 10 years.
So your company, do you produce just the catalyst or you produce the biodiesel?
We have a pilot plant to make biodiesel, but only to showcase our catalyst technology. We do not want to make biodiesel, even though the biodiesel we made, we actually 50% donated to the university. They have a biodiesel bus system. The other 50% we sell it to REG. So they take our biodiesel. And we're just using that pilot plant as a demonstration of our catalyst technology.
I was just kind of wondering, does the biodiesel you make, do you have transportation problem? A lot of biodiesel have transportation problem.
We, right now, as I mentioned, it's been distributed immediately on our site. And the glycerin, we're actually selling the glycerin. The glycerin, because ours is actually salt-free. So our glycerin is actually quite, quote-unquote, hot for some of the personal healthcare people to come in and use it. And so we are actually making good revenues from that. Yes?
So what is the cost to produce the methylferrous nanoparticles?
Good question. Good question. Well, that's a trade secret I cannot tell you. Because we still need to sell it to people. If I tell you how much it costs, people will say our profit margin's a little too high. So I'm not going to answer that question. Okay, so, but all I can say is this. I mean, we are actually price competitive. In fact, we're selling now the T300 for the transesterification reaction. That catalyst, we're selling it exactly the same price as sodium methoxide. So that tells you how cheap we could get it down.
What other applications do you envisage for this kind of chemistry?
So anything you can imagine to do transesterification reaction and esterification reaction, we could use this as a solid catalyst. So in fact, we are talking to several companies that are not biodiesel producer. They are actually interested in making personal healthcare fatty alcohols. But in order for them to get that, they typically convert the oil to biodiesel first, then do the reduction. So we are actually talking to those that are using that.
So a lot of people use them for alkyds and that kind of thing as a starter, but you have to use much longer chain alcohols and acids to do that instead of the short chain. What happens when you increase the length of your starting chemicals? Does the technology work just as well, or do you have other things? Do you see it?
The reason why those always becomes an issue is that the catalyst leaching. Because depends on your chemical input, right? Obviously that the more soluble these catalytic sites can be leached out in those reactants, then that becomes an issue, right? In our case, we are leaching, it's actually at the PPB level because it's all embedded inside of the structure. So we have not encountered that. With one project that actually people are interested in using isobutanol as one of the feedstock. And I cannot tell you the application, sorry. The NDA. Okay. Yes?
So actually in one slide you show that it's not the catalyst, it's a nanoparticle structure. So it shows that it absorbs like small carbon chain molecules.
Oh, you're talking about the sequestration particle.
Right.
Okay.
So my question is, have you ever thought about using this structure for like oil refining? Say because it can absorb like small molecules, which are like very important for oil industry. So I'm just wondering.
Yes, we have. Yes. The short answer is yes, we have. We're actually now in conversation with 2 petroleum companies about that. Exactly that. But again, sorry, I can't tell you the target.